Hello, welcome to scGRNdb. This is a functionally annotated cell type network database. And right now we're here on the home page. We're met with the workflow figure of scGRNdb, which includes 4 different functions: network browser, network prioritization, network comparison, and network pathway annotation. If you scroll down, you can see an overview of some general stats of all of our networks, including the total number of networks split by tissue, cell type, as well as a distribution of node and edge numbers per network, as well as a description of all of the atlases we've used to curate these networks. If you're interested in how we constructed the networks, we used a method called scGRN developed by our lab, which you can check out even more at our GitHub page if you're interested in running those yourself. So let's first get started with the network browser tool to visualize networks. If you click the Explore Networks tab, you're going to be met with the network analysis page where you can run any of these different functions on scGRNdb. If you come down, you can actually click on these network boxes for more information on each of these tools. For network browser, it's pretty self-explanatory, just to visualize and download any scGRNdb network. So I'm going to click Browse Networks, and here I'm immediately met with the table of all the networks that are in the database. You can search by different criteria like the species, atlas, tissue, and cell type that you're interested in. You can also search on this search pane as well. Here I'm going to just focus on a liver network, particularly the hepatocyte network. You can select the network by clicking on the table row, and you can also download the network, the modules of the network, as well as the pathway and disease annotation files. And you can download all of them in a zip file here, as well as on the sidebar here. Whenever you click a network, it'll pop up on this selected network sidebar, and then you can click Add to open up the visualization of the network. You're brought to the visualization tab in the network browser. This overall network figure is showing all of the modules that are in your network, as well as the connections that are between them. So each node here, right, is a collection of highly connected genes, and they can have connections to other modules as indicated by these edges here. If you click on any of these modules, you can see the module annotations for them. And now if you click on pathway enrichments, you can explore any of the pathway and disease gene databases for information on the enriched pathways. You can also adjust the size of the figure to suit your needs and then download it as a PNG. Now, if you're interested in visualizing these modules, you can actually click on Load Module Network here at the bottom, which will take some time to load all of the genes that are in that module. This module is quite big and there are a lot of connections between them. So there are a couple of ways to interact with this visualization. One is to remove the more peripheral nodes and focus on what's most highly connected in the center. And that's by using something called a node degree filter. As you see, if I look at the top 10% of nodes highly connected, as well as their neighbors, this is what you see. You can also decrease it to the top 1%, right? Refresh the layout, and you get a much clearer picture of what's the most central in the network. You can also filter by edge weight as well, which will kind of remove the low importance edges based on a certain percentage or proportion. And you can always refresh the layout as well to try to clean up the arrangement or the layout of the network. If you want to export any visualization, click the Export Visualization, and there are 2 options. One, you can download the complete network as a PNG, or you can download the JSON object and load it into a Cytoscape desktop to customize the network even more yourself. You can also convert the gene annotations from human to mouse using this conversion button here. It'll take some time to load all the conversions as each gene is fed through the conversion API. But once that is done, you can use that. And this is especially important if you want to compare networks across different species. So this is one way to view the genes in the network if you don't have any prior gene sets you want to focus on. A great way is to click on modules that are interesting based on the biological annotations and load those module networks. If you are interested in certain genes, you can either input a list of genes or upload a gene file yourself. Here I'm going to focus on these 4 genes. Click Find Genes, and inside the module view, the modules that contain those genes will be highlighted in yellow. You can kind of, again, explore the information on those genes. Then you can also head over to the Search Results tab to view the actual network surrounding your genes of interest with them highlighted in yellow. Again, you can convert to mouse if you'd like. Now, you can also increase the neighbor search to look more deeply into the network, and again, export the visualization as needed. Now, if you'd like to visualize other networks, you can always X out and remove the visualization by Xing out here, as well as Xing out on this visualization here. You can also deselect this table. So if I click this and go back to visualization, you see that it's not there anymore. So that is it for the Browse Networks tutorial. Next, we'll head on to explaining how to use the network prioritization.